psilocybin mushrooms of the southeast. It's about how little we know about mushrooms, even the ones we pay attention the most to. Hopefully, that's something that will be conveyed and come through during this talk. So, in the southeast, we have, I'm going to call it 3.5 different genera. We've got Psilocybin, Peniolus, or no, 4.5. Psilocybin, Peniolus, Gymnopolis, Pluteus, and Gallerina with an asterisk. So first I'll talk about the Psilocybes, everyone's favorite psychoactive genus. This one probably needs little to no introduction. Psilocybin cubensis is the one most people are cultivating at home. But here in the southeast, it does grow wild on horse manure as well as cow manure and potentially other ungulate manures. It was originally described by Franklin Sumner Earl in Cuba, hence the name cubensis of Cuba. It's been described a number of times since then by other people who just were not aware that it was already described. For instance, William Alfonso Merle described it from Florida as Stropharia cyanescens as well. Interestingly, Merle's collection came from sawdust rather than manure, which is kind of interesting. And it makes me wonder if it was actually cute, but Guzman decided it was the same thing. Alright, so this is a distribution map according to iNaturalist of Psilocybin cubensis in the southeast. So you can see, covers all of Florida. We've got it in eastern Texas, Louisiana, and apparently it's been documented as far north as southern Missouri. So that's pretty neat. Here's some nice photos of Psilocybin cubensis in habitat. This guy and this guy on the left were in cow manure. The guy in the top right was found in horse manure. And then here's one grown from cow manure, and you'll notice the indicative blue bruising on the flesh where it's damaged. That we're all familiar with. And then, like I said, this one is extremely popular to cultivate at home, and as you can see, it'll produce these large flushes of mushrooms, which lends itself to being an ideal candidate for the black market. They can get really big in cultivation as well. You can see here this, I believe this is a Melmac. It's larger than my hand. And then it occasionally can take on these strange moraine or fan-like morphologies. This one is the Enigma, photographed by Alan Rockefeller in Mexico. And then we've got Psilocybin tampanensis. This one is also somewhat popular in cultivation as well, but seems to be extremely rare in habitat, or at least we don't have a lot of people that are getting to a species level identification with them. Likely because it's just another little brown mushroom. It's very non-distinct. It's not as charismatic as Psilocybin cubensis in the wild. So these photos were of cultivated Psilocybin tampanensis, though, and I don't think I included it, JT here found one in southeast Georgia on his cow pasture, and it was probably the most abundant fruiting I've ever seen pictures of in the wild. It was pretty impressive. But yeah, this species is also known to produce sclerotia, or these underground masses of dense mycelia that function as almost like a dormant form of the fungus, so that when the conditions are right, it can fruit again. But interestingly, the sclerotia have never been observed in habitat. They've only been observed through cultivation. And something I've noticed whenever I've cultivated these in a legal state was I would plant the sclerotia and then I would have it fruit. But by the time it fruited and I went to go dig back up the sclerotia underground, they were like this like dissolved, juicy mess. So I suspect that we may never find the sclerotia in habitat. I feel like they start decomposing as soon as it starts fruiting. The only way I can see us finding it is maybe just by like looking for soil samples and just stumbling across it like a needle in a haystack, and it just hasn't fruited yet. Yeah, so far it hasn't been found in a while. And then, so this species was described by Stephen Pollock out of Tampa, Florida, hence the name Psilocybin tampanensis. For those of you who don't know, Stephen Pollock was a very interesting, eccentric mycologist who lived in San Antonio, Texas, and Hamilton Morris wrote this article for Harper Magazine called Bloodsport that documents pretty much his whole life, and he was known for doing a lot of interesting experimentations, like giving the sclerotia of Psilocybin tampanensis to children with autism and claiming that it cured it. And then shortly after that, he was assassinated by the San Antonio Police Department. And there's all this sort of like conspiracy surrounding like why he was assassinated. It is worth noting he was also a licensed pharmacist and was basically a legal drug dealer that operated out of his house. So it likely wasn't just the mushrooms that got him assassinated. It's worth taking a look at this article, Bloodsport. And then, so here's the iNaturalist distribution of the species. This certainly isn't complete, and this goes back to what I said about it being difficult to identify as a species, but it's also a matter of people not posting to iNaturalist. That's something I'm going to hammer in throughout this talk is any mushroom you find, especially the interesting ones. We probably don't need more turkey tails on iNat, but if you find an interesting mushroom, before you put it on the Facebook group for that instant gratification identification, put it on the iNaturalist so that that data means something long term. Because everyone knows, as soon as you post it in the Facebook groups or the shroomery, that post gets buried very quickly, and it's very difficult sometimes to resurrect that data. And when you put it on iNaturalist, it becomes a part of this larger data set that people like me who are giving talks utilize for their distribution maps. According to iNat, it's been found in South Carolina, a couple around the Atlanta area. This will be JTs from Southeast Georgia, and then we've got one in Louisiana. And then there's also one in Texas, but if I recall, I looked at that one and I was a little skeptical about whether or not that was Psilocybin. And then here's some examples of them in cultivation in a legal state. But in this instance, in the lower right, I planted the sclerotia into this, 12 tray, or 12 cell tray, and I kept them moist, and a couple days later they fruited. And then with that same batch, I took the colonized grain and bulked it into a separate pot and got fruiting bodies that way as well. And then here's an example of the sclerotia, or what they call in Amsterdam magic truffles. These are slightly dehydrated, but you can see I don't have a scale bar, unfortunately, but they can get pretty large. I have a buddy who allowed his tampanensis to colonize the grain for, I believe it was 14 or 16 months, and after he took it out, it was the size of like a little Nerf football. It was like that big. I don't know how much it weighed, but it was pretty impressive. Alright, so our next philosophy. This one is, assuming it actually does occur in the southeast, this is the rarest philosophy we have down here. So Psilocybin mammalata was described by William Alfonso Merle from Jamaica. And exactly 10 years later, Harry Tiers, who, if I'm not mistaken, was a West Coast mycologist, he was visiting Florida and went to the Highland Hammock State Park in south central Florida. Beautiful old growth forest. If you guys ever visit Florida, that's like great spot for biodiversity. And I guess there's incentive to find a cool psilocybin there. But he claimed to have found it in August of '58 in Highland Hammock, and determined that it was the same Psilocybin mammalata that Merle described from Jamaica. I believe in Jamaica there was about 3 collections made, totaling maybe like 6 or 7 fruit bodies, maybe 8, and then I don't know how many fruit bodies were found by Harry Tiers at Highland Hammock, but it's like 2 or 3. So maybe only about 9 fruit bodies in total have been ever found of this species in history. But fast forward to August of 2023, just 40 minutes away from Highland Hammock State Park in Yeehaw Junction, Florida, again, like southern, central, like middle of the state, around Lake Okeechobee, Florida. Dr. Matt Smith, the mycology professor from University of Florida, him and his lab were doing a fungal biodiversity survey at it's called the DeLuca Preserve. It's a private preserve that, to my knowledge, only University of Florida has access to. But he found a psilocybin there that macromorphologically and microscopically seems to align with this species, Psilocybin mammalata. Now, this collection was DNA sequenced, and it does fall in section Cordyspore, the section that mammalata falls in as well. But unfortunately, at the moment, we don't have a type sequence for the holotype of Psilocybin mammalata, so we can't match it genetically. But I'd say with pretty high confidence, this is likely Psilocybin mammalata, and what Harry Tiers found in Highland Hammock was as well. Alright, so this is the newest psilocybin that's been described from the southeast. This is Psilocybin niveo tropicalis. Niveo meaning white or snow white, tropicalis meaning tropical, so it's kind of like tropical snow. And as you can see here, we have like this really pale, almost white colored Pilius and stipe that, in contrast with the typically dyed mulch that you find them in, looks almost like scattered snow in the mulch. So I felt like that was an appropriate name. Me and my buddy Andrew Chambers, we discovered this mushroom back in April of 2019. And I made a bunch of collections of it over the course of like 4.5 years in South Florida. Interestingly, it's only been found in one county in South Florida, Palm Beach County, and most of the collections were made in a town called Jupiter, Florida, which me and my buddy are from. And finally, in March of last year, so a year ago this month, myself and Joshua Bergback was a tremendous help in putting this together. He had the taxonomic and manuscript writing abilities and was able to contribute greatly to this publication. But also Alan Rockefeller and then George Jacobs, who did some HPLC analysis on this species, contributed to this as well. So now this is an officially named species. And you can, if you want, you can check out this publication in the journal Macalvania. It's an amateur mycologist journal that is available through the NAMA, or North American Mycological Association's website if you're interested. One really interesting thing about this species, we got 5 different gene regions sequenced for this paper, and Josh threw it in the tree to compare it with all the other known Psilocybin species. And it comes out in this clade that most closely matches a lot of Asian species. Excuse me. For instance, we have Psilocybin Thai duplicatus cystidiata, Psilocybin Tyrogonio maculans. Those 2 species were described from Thailand. And then it's also pretty closely related to Psilocybin wayanadensis, which was described from the Wayanad district in India. So it sort of begs the question, given its sort of artificial habitat, these irrigated mulch beds that they're found in, was this thing imported from somewhere else? If so, how long ago? But also, just because it is most closely related to these Asian species doesn't necessarily mean it's from Asia. But it is something that you can't help but speculate when you see this. And then you can also see its sort of sister to Psilocybin ovoideo cystidiata, which is another one we do have here in the southeast. And then Psilocybin cubensis and Chuzion. Actually, I'm not going to attempt to pronounce that one. Chuzion something along those lines, which was described from China. And this whole little section here represents what's called Psilocybin section caerulescentiens. And then here's the iNaturalist distribution report. So again, this has only been found in Palm Beach County, which at the northernmost point, Jupiter is the second furthest north town in Palm Beach County, and then down here, just sort of south of Boca Raton, is where Palm Beach County cuts off. And you can see all of the observations fall in that range, with a high concentration being in the Jupiter to West Palm Beach area. But this is really cool. So under 365 nanometer UV, the species fluoresces this like raspberry, pinkish, almost red color, which in combination with the blue bruising and the white coloration, it feels like a very American species, the whole red, white, and blue thing going on, which I think is really cool. And then another really unique thing about this species is on top of the typical spore morphology you'd find in this section, which is like rhomboid, subrhomboid, which you can see here. It also has these, what Josh taught me is called apically cleft or heart-shaped spores, which isn't totally unique in mushrooms. Occasionally you do get these mutated spores in other species, like I know there's some Mycena it's been documented in, but it's extremely rare, and it's like 1 out of every 10,000 spores in other species. But in this species, for some odd reason, it represents like 10 to 15% of the spore mass is this unique, like apically cleft spore morphology. Alright, so the next species we've got Psilocybin caerulescens. Is there anyone here familiar with this species? Yeah. So yeah, this species was described originally from Alabama by William Alfonso Earle. Additionally, Paul Stamets found this species in Georgia and decided to call it Psilocybin wileyii, naming it after his friend Andrew Weil. But it was later determined to be synonymous with Psilocybin caerulescens, and there's a little bit of a debate about this and whether or not it's a legit species, but it's pretty. ITS sequences matches, microscopy, macromorphology, it all matches, so I think it's the same. And Yeah, these were all photos taken in the Atlanta, Georgia area. There's really dense. Actually, I shouldn't say there's a dense population of this, but there's a dense population of humans that tend to look for this species in the Atlanta area. So when we get to the distribution report, you'll see there's just like this huge cluster of them in Atlanta. But this species also occurs down in Mexico as well, and I'm sure Dusty, you've seen it down in Mexico. Have you seen the species in Mexico? No? Just the zaps? Well, Alan has plenty of photos from Mexico, but this is the distribution in the southeastern United States. So it goes as far west as Louisiana. We've got an observation in Mississippi, Alabama. Again, this huge cluster around the Atlanta area, some in South Carolina, and then the one in North Carolina, I believe, was only found in the last year and a half or so. And it looks like there's one maybe right around this area, Chattanooga. But about, what was it? I think back in the fall, someone found it in Arkansas and posted it to Facebook. And so, again, I'm going to reiterate this. It's really important that you put any and all interesting things you find on iNaturalist so that it can be included in this larger data set. Yeah, it was, posted to Facebook from Arkansas, and I tried finding it again before this talk, and again, just got buried in one of these Facebook groups. And these groups are fine for getting these quick instant gratification identifications, but you really got to put it on iNat. All right, brings us to our next one, Psilocybe ovoideo-cystidiata. This picture was actually taken 2 or 3 days ago, again in the Atlanta surrounding area, growing out of mulch in a community garden. I was actually down on the ground, photographing all these psilocybes, and there was some group of elderly people walking around, enjoying their day in this community garden. And they went, "Oh, what are you looking at? What is that?" And
they were just afraid you were taking their stash.
Sorry?
They were just afraid you were taking their stash.
Yeah.
You found them.
But I told them what it was, and they were like, "Oh yeah, my nephew found some of those in my yard." And so they were very much aware of what it was, which is pretty cool. And they were very cool about it. They weren't judgmental.
They harvested.
Sorry?
They harvested after you left.
There was plenty to go around.
Oh, nice.
Yeah. By the way, if anyone has questions, feel free to speak up at any point. All right, and then this is a nice little cluster photo taken by my buddy Efley Shroom. That's his iNaturalist and Instagram handle. And then more clusters of it, but they don't only grow out of these wood chip landscapes, but they also will grow directly out of box elderwood, heavily decomposed box elder wood, which is really cool. And I don't have the picture here, but
is that photo on the bottom left a leaf that it's coming out of? I'm so confused. They look like it's looking through a portal. What's going on there?
This is wood, and it looks like the mycelium has rhizomorphically attached itself to the leaf, and so the leaf is sort of sticking to the wood.
Oh wow, that is an amazing shot.
Yeah. Yeah.
Was this just a couple days ago too?
No, this was definitely in the last month or so, and yeah, these were taken by. His name's Justin. He lives in the Atlanta area. I call him
when Jordan came, that's who he met up with.
Yeah, he's like the Psilocybe whisperer of Atlanta. He knows.
And he's taking Josh out too.
Yeah, he's great. Really cool guy. And then this is the distribution report for Psilocybe ovoideo-cystidiata. So I'm going to refer to him as ovoids for now, because that's a mouthful. cool. But ovoids were described only, I believe, in 2008, if I'm not mistaken, by a guy named Richard Gaines, and it was described from Pennsylvania, and in the Northeast and into the Midwest, like Ohio, you'll find them in these what are they called? These like floodplain-like environments coming out of like buried wood, as well as the landscape mulch beds. But these like floodplains seem to be its like natural habitat, and I forget the guy's name, but someone who claimed to be like the Johnny Appleseed of ovoids apparently took like a bunch of like 5-gallon buckets of the colonized mulch and brought it over to the West Coast, into the Pacific Northwest, and down into California. And so it's been spread throughout the West Coast into these like artificial landscaping beds. So now it has this like disjunct distribution that just totally skips over the middle of the country. But you'll see, kind of starts up in the Northeast, as far north as apparently Vermont, and just runs down through the Appalachians. And see, the southernmost point that it's at least been observed is this Atlanta area. But I suspect it's probably in Alabama and maybe even Mississippi as well. And it's just a matter of people, more people going out there and looking for them in less densely populated areas and posting them to iNaturalist. All right, so the next genus we're going to talk about is the genus Panaeolus, or the mottle gibbs. So, probably the most common in cow fields, Panaeolus species is Panaeolus cyanescens. So this is extremely potent, potentially the most potent Panaeolus species that we have in the southeast. And it grows on cow manure. I've personally never found it on horse manure, which is interesting because I've definitely observed Psilocybin cubensis on the horse manure, but I've never found this species of Panaeolus on horse manure. I found other species, but for whatever reason, this one seems to be strictly associated with cow manure, at least here in the southeast. But it's extremely common, especially in South Florida. It's still pretty common further north, like central, northern Florida, Alabama, and southern Georgia. But I mean, it's almost more abundant than Psilocybin cubensis in South Florida for whatever reason. I think it just really likes that like truly tropical climate. But it was actually originally described from Sri Lanka on the other side of the world as Agaricus cyanescens, and it's distributed sort of cosmopolitanly, like around the equator, like all around the world. So it can be found pretty much almost any continent you're on. Maybe not Antarctica, but every other one. Here's the distribution report for it for the southeast United States. You'll notice we've got a couple outliers in the middle of Virginia and then eastern Oklahoma on the border of Arkansas. Which is really interesting to me. They must have had like a really warm, wet summer that year. But for the most part, you can see it's Florida, all over Florida, eastern Texas, Louisiana. A lot of overlap with the distribution for Psilocybin cubensis here, and it shares the same substrate. So you find one, and likely you can find the other. And then here's some photos of it in cow dung. Got a little scale bar there for size reference. They can get pretty tall and lanky depending, like if there's a lot of like grass, kind of like almost overgrowing the manure. They like growing up like through the grass and peeking their little caps out for humans to find, of course. They can get pretty tall. All right, the next Panaeolus species got is Paniolus cincellus. This is probably the most widely distributed psilocybin-containing mushroom on the planet. Found in every state in the United States. Found every continent in the world. Again, probably except Antarctica. And yeah, this is a grassland species, and it tends to like these. What's the word I'm looking for? Like high organic matter, very rich lawn landscapes. You know, if you go to like a newly constructed housing development and they have like freshly laid sod everywhere, you're gonna find these, or community parks where they freshly laid sod. You're gonna find these.
Those are different species.
Sorry?
We're sure those are different species, the cincellus and the blue meanie.
Oh, and Paniolus sinessens?
Yeah.
Oh, yeah, without a doubt. Yeah, macromorphologically, microscopically, genetically, it's...
Totally different.
The thing about Paniolus is there is a lot of macromorphological overlap. They all really...
They look similar to me.
Yeah. And you know, what complicates things even more is oftentimes you'll have fresh sod laid on like composted cow manure from like the bagged cow manure from the store, and you'll get things like Paniolus sinessens and Psilocybe cubensis coming in the lawn as well, and so there can be this overlap of habitat. But the Paniolus sinessens, the flesh bruises like extremely strongly, and it's actually like somewhat rare to see any visible blue bruising on the cincellus. You'll usually find it at the base of the stipe where you start seeing the mycelial kind of rhizomorphs. And if you get lucky, you'll see a little hint of blue bruising there. But the sinessens, like the whole thing will just turn black, you know.
Has Julian crossed the two? I have so many
Uh,
questions.
No, Julian doesn't really work with Paniolus, but I mean, I would be surprised. These are actually like pretty genetically distinct. They're not in the same subgenus. And then, yeah, again, this thing is everywhere, even up in Alaska. I'm sure if I, expanded the filter to all of North America, you'd see it all throughout Mexico and Canada. Yeah, it's everywhere. All right, so this is, if not the rarest Paniolus species on the planet, it's tied with maybe one or two others. This is Paniolus chlorocystis. It was described in 1978 by, if I'm not mistaken, what's his name, Roger Heim, as well as Stephen Pollock, the fellow who described Psilocybe tamponensis. This one has a slightly interesting story. So actually, Stephen Pollock, when he found Psilocybe tamponensis with Gary Linkoff, he had skipped out on this mycological conference because he thought it was too boring. And he was hearing rumors about this new bluing Paniolus that was being found in South Florida. And this mycological conference was taking place in Tampa, Florida. And so Stephen Pollock was actually, he had heard rumors about here, and then some microscopic features, including these like metalloid cystidia, which is what's the word? Synapomorphy, is that the right word? It's a synapomorphy, or it's like indicative that it belongs to the subgenus Copelandia, which this group of Paniolus species, these like deeply blue bruising species, used to have its own genus that was known as Copelandia, but then Gerhardt moved it and combined it with Paniolus, and now it's just a subgenus. But that was the only picture we had until I came along and I looked at the paratype, and now we have a much better picture of the thing. So yeah, this is what we're working with. When you go into these fungariums and you look at these old collections, this is for the most part what it looks like. But one cool thing is I was able to put these guys under the microscope and get some really high quality micrographs. So now we have some really good micrographs, and So these are the metalloid cystidia I was talking about. It's these like thick walled, like what's it called? Not mucronate, but beak shaped. It's got a sort of beak shaped apex at the top, and they're just throughout the entire, like all of the gills, the entirety of the gills. They're really pretty. They can even be beset with these like little crystals that are really cool to see under the microscope. Really beautiful structures. Now, I actually think it was in May of last year. I was actually able to go out and collect this mushroom in habitat, and it took an extremely long time to find even a single fruit body of them. I was combing through the grass in this like it's funny, it's actually one of the neighborhoods that Psilocybin niveo tropicalis grows in. About 20 feet away under an oak tree in the grass, I found this mushroom in this neighborhood. So two really rare psilocybin containing species. Same neighborhood within 20, 30 feet of each other. And I was able to find a collection of it, and took some really nice photographs.
Gorgeous photos, Scott.
Yeah,
So nice.
thank you. And then I might have slipped a gill into an envelope and sent it to a friend, and he cultivated them, and they lend themselves really well to cultivation, just like Peniolus senescens or Peniolus cinctulis. You can see here, they create these like fat clusters. And very soon, if it doesn't get through the grapevine in another way, I'll be making spore prints of these available for the interwebs. So hopefully more people can cultivate them. And then here's the distribution report for them. I ended up finding them 3 times in this Jupiter area. And then here's actually Alan Rockefeller's observation of one of the paratypes. So that's not a modern collection over there. And then I found another observation on iNaturalist that macromorphologically resembled this species in the Boynton Beach area, and I decided to call it that because it didn't seem to align with anything else. So I'm pretty sure this other iNat user found it in that area. So again, another species that is strictly found, actually I take that back, because the first collection of this was found in Miami, but still, all the modern collections of this are all in this Palm Beach County area. I feel like, and I'm biased because I'm from Palm Beach County, but I feel like it's like a vector for very special psilocybin-containing mushrooms. All right, so this is the newest kid on the block in the genus Peniolus Peniolus. This is Peniolus oligotrophus non-prov. This is an unpublished species of Peniolus that up until a couple months ago had only been found in my yard. I was out in my yard and I found these Peniolus, what I thought were Peniolus cinctulis at first. I was like, oh wow, I've got Peniolus cinctulis in my yard. Surprise, surprise, it's everywhere. But then I got down on my knees to worship them, and I noticed that a slightly different coloration from what I was used to seeing for cinctulis. And on top of that, I know that cinctulis grows in these sort of enriched landscapes. And my yard is a sandy, degraded, like white trash looking yard. So I figured it was a little odd that it was growing in this organic matter devoid sort of habitat. But in any case, I decided to try them. I still didn't think they were different from cinctulis. I was like, I kind of dismissed the ecological difference, and they ended up being active. I only ate a small amount, but I got that little buzz going that was reminiscent of any other time I'd eaten low doses of cinctulis. But just for the hell of it, because this is just a habit I have. I took photos, I dried part of the collection, and I sent it to the Ohio Mushroom DNA Lab to be sequenced. Now, simultaneously, Jason Slott was soliciting for spore prints of Penniless cinctulus, and I was like, "Oh, Jason, don't worry, this was planned for comedic relief." But I was like, "Hey, Jason, I just found some Penniless cinctulus in my yard, or what I think is Penniless cinctulus here." So I dropped him a spore print, and he wanted to get a full genome out of this species. So I sent him the print, he cultured it, and simultaneously, or like within a week, I got the first ITS barcode results back from Ohio Mushroom DNA Lab, and then like within a week, Jason Sly had produced an ITS sequence for it, and then eventually a full genome, and they ended up being pretty far away from cinctulus, and actually these are genetically closest to Penniless phoenisecii, which is an inactive species of Penniless. Which is really interesting. And also, phoenisecii is also microscopically unique. It's the only Penniless species that has like light brown spores in mass on a spore print. Most Penniless, they do have like under the scope like light brown or really medium brown to dark brown spores when you look at them under the scope. But when you take a spore print, it appears as jet black. Phoenisecii is different, where it has like really light brown spores, and when you take a print, it's more like a rusty dark brown coloration, and they're like highly they're very coarse. They have this like texture on the surface, where every other species of Penniless has these smooth, darker brown spores. Here's a couple more, but you can see. So the cinctulus tend to be more like sort of on like a grayscale in coloration with a black banded look or near the margin. These are sort of like a, I don't know how to describe that shade of brown, but they're on like a browner to like, they almost get this like black coloration on the cap as well. So, going back to your comment, Dusty, with cinctulus and cyanescence looking very similar. These things macromorphologically are just, a lot of overlap. They all look the same. There's just these subtle coloration variations.
You pulled out the psilocybin gene cluster and looked at that?
Yeah. So once we did get the full genome, Jason Slott was able to pull out the psilocybin gene cluster, and that will be mentioned and compared with other gene clusters in the paper that we describe it in.
Did you do a chemical workup?
What?
Did you do chemical workup?
Like HPLC?
Yeah.
No. So, well, depending on the timeline, so I actually recently sent some spores to my I have a designated Penniless cultivator on the West Coast who goes by the Pandalorian, and he only grows Penniless species. So he's my guy, and he'll grow active, non-active pans for me. When I find these rare collections of Penniless that only have like one fruiting body, I'll take a gill, or if there's a spore print, and I'll send it to him so he can cultivate them and produce many fruit bodies so that I can study them without, what's the word? Just losing more material from the limited material I have, which is really nice of him. And he's all pumped up about it. He's excited to be a part of the research. So anyway, hopefully he'll get this to fruit, and then we can send it to someone like Jordan Jacobs to do some HPLC analysis. And then I mentioned earlier, up until recently, it was only found in my yard, and last year, I think around October, my buddy Aiden, who's in Southwest Florida, found it in like an actual habitat. So he found it in this like pine flatland, extremely sandy soil. So again, like the ecology like kind of matches my lawn, like just really sandy. Okay, there's a couple more photos of the habitat he had. I guess this is the only one I included. But you can see some pine needles here and a little bit of cypress. But you can see, there's no grass, no weeds. It's just like straight sand. And then here's some of the microscopic features of this species. So again, you've got these like smooth, really dark colored spores. You've got some nice stout basidia here, some little fatties. A lot of basidia. Some of these mushroom species can be like lanky, like flexuous, almost flaccid-looking basidia, but they're nice and plump. And then on the left-hand side, we've got the Psilocybin, or these microscopic cells just along the edge of the gill. They're really cool. So again, yeah, this one turns out to be closest to Paniolus phonosectia, and you can see Paniolus cinctulis is, in a whole other group seemingly relative to this one. And then here's the distribution report. So up in my yard and then down almost in the Everglades. So, again, this goes back to Dusty's comment about them all looking the same. I'm sure there's other observations of this thing on iNaturalist that have just been identified as Paniolus cinctulis because it's so difficult to separate them out macromorphologically. So this is a call for more people to just make iNaturalist observations of everything they see, collect them, dry them out, send them into get DNA sequence, and I'll talk a little about that at the end, how you can go about doing that. Because without a doubt, it's not like this thing is only occurring in my yard and Naples, Florida. It's got to be everywhere else. So, okay, this is misleading, potentially. So, the next species we have is Paniolus bisporus. And this is a photo that Alan Rockefeller took in southwest Florida. And he did some microscopy on it and determined it was Paniolus bisporus. And this was based on the fact that he only saw two-spored basidia, which is what it is named after. So macromorphologically, this one is definitely closest to Paniolus cyanescens, except it grows in a more cinctulis-like habitat, these enriched lawns. And unlike cyanescens, it strictly has these two-spored basidia. But here's the thing, Paniolus cyanescens can have either four-spored or two-spored basidia, but it's more common to find the four-spored ones. And when Alan took a look at this, he only saw two-spored basidia. But just 3 days ago, I got ITS sequence data back for this specific collection of Alan's, and it actually ended up matching Paniolus cyanescens. And so, and I just sent it off to Jason to get full genomed as bisporus. So there's a chance Alan just didn't see any of the four-spored basidia. So I'm going to have to double check this collection and see if it has four-spored basidia. If I do find the four-spored basidia, it probably is cyanescens. If it doesn't, it's going to be a lot of work figuring out what the heck this thing is. But this collection aside, Paniolus bisporus does occur in the United States, and excluding this Florida collection, has been found in at least South Carolina. So this is another one. We need more people to get out there and, comb through the grass for 6 hours at a time and find these things. All right, so this is another extremely interesting one. This was found, again, in southwest Florida. So this is, tentatively for now, what I'm calling Paniolus cylindrosporus. So this was found 7 or 8 years ago by my buddy Andrew Kunick, who I'll talk about a little more later in this talk. He found this really odd-looking Paniolus species in southwest Florida on a lawn, he managed to take some spore prints of it and ate the collection. So he didn't save any of the original fruit bodies, but he ate the collection under the guise that it was just, penniless cinctulus or something similar. And he did report that it was active in man. So fast forward, now that he's a more nuanced, literate mycologist, he went back and was like, dang, that was an interesting collection of penniolus, and he managed to dig up this 7 or 8 year old spore print, and he was able to culture it and fruit it, and then look at the spores, and we went, holy crap, these spores are unique. They do not match cinctulus or anything close to it. So he did a full microscopic workup, and together with me, there's this guy in Italy, this Italian mycologist named Pietro Boto, and he's an expert on the genus Penniolus, and he decided this lined up perfectly with a species that was described from Germany as Penniolus cylindrosporus. Now, we got a sequence from this one, but Penniolus cylindrosporus does not have a sequence yet. It's one I requested on loan recently, so hopefully I'll have it at the university in Florida here soon, and I'll be able to DNA sequence it and get it to match up with this collection. But
What do you think of that one all the way to the left of that photo?
So yeah, this is definitely a mixed collection. The one on the far left is likely Penniolus phonosecii. That's definitely a mixed collection that Andrew had. But again, this is 7 or 8 years ago. He was probably like 14, 15 at the time. He wasn't the refined mycologist that he is now. All right. Speaking of Andrew Koenig, the next genus we have is the genus Gymnopolis. We have no good names for any of the Gymnopolis. What you think is a good name for a Gymnopolis species is not. My buddy Andrew is currently doing a type study on the genus Gymnopolis, specifically of the southeast. And so what that means is he's essentially looking at all these collections that were described about 89 years ago by William Alfonso Merle, who described most of the species in the southeast, including like 7 or 8 species of Gymnopolis, and about 4 or 5 of them being active. And so what Andrew's trying to do is he's trying to attempt to resurrect some of these old names that have been forgotten about, that are no longer used. So right now what we do is we sort of blanket or just umbrella like some of these names like Suspectabilis or Junonius or Dolepis, and we kind of just apply it to all of the blue bruising Gymnopolis species that we have. But likely, we have no idea what any of them are. All of the Gymnopolis experts died probably in the 70s, and so there hasn't been one until now with Andrew Koenig. So, because I cannot present any species, I'm just going to present, we already talked about Merle. This is Mr. Andrew Koenig. Like I said, type study on Gymnopolis, resurrecting Merle's names. He is a bit of a character. But yeah, this is what he's kind of working with here. He's looking at these almost 100-year-old collections. You can see this one from April of 1938. And he's doing microscopy, as well as successfully DNA sequencing some of these mushrooms. And Merle's collections are notorious for not being able to get sequenced. All the, Alright, the next genus we've got is the genus Pluteus, or what we call the deer mushroom. This is potentially Actually, I hesitate. I was going to say the least understood genus as far as its active species goes, but there are a couple other ones that compete for it. But definitely less understood than the Penniolus or the Psilocybin species. So the first species I've got going is Pluteus septocystidiatus. You know, it's names like these that really beg for a common name. So you can. It's probably hard to see. Dang it. I thought I had another photo of that one. I have another photo somewhere with a close-up of the apex of the site where it meets the gills, and that's typically where you see bruising with the active Pluteus species, at least before you handle it. So even before you bruise it and handle it, usually there's a faint blue ring, like a little halo around the site where it meets the gills that tends to tell you that it's active. And then, you know, start picking it up, handling it with your paws, you know, it starts bruising. So this one, this is another example where we have these like disjunct populations. I have found them in a couple places in Florida. I know I have a buddy on the west coast of Florida who's found it. And then we have this large gap skipping the rest of the southeast. And, you know, we've got Ohio, Indiana, Iowa, what looks like New Jersey over there. But this isn't a reflection of where it grows. This is a reflection of how few people are able to get it to species level resolution on iNaturalist. And, you know, part of this is definitely lack of DNA sequencing some of the collections here in the southeast. Again, this is another genus just like the genus Peniolus, where macromorphologically they all look the frickin' same. It's so hard to tell the difference between them. And even some of the world Pluteus experts are unable to tell them apart macromorphologically, microscopically. The only way that you can tell them apart is with the genetic information. But yeah, here's a couple more fruit bodies, and then I clumsily squashed the spore print, so you have a bunch of collapsed spores. They should be like perfect like globes or spheres, but you can see these are some collapsed Pluteus spores. Alright, so this one has what's called a Cryptonomum temporarium, or a temp code. So this is called Pluteus species FL05, at least for now. So this is a species that my buddy John Cook found in central Florida in a very unique old growth forest kind of environment that kind of blurs the line between the tropics and subtropical climate. And this one came out not matching anything. So there were a couple DNA sequences in GenBank from South America labeled Pluteus harrisii. Now, Pluteus harrisii is actually described, again, by Merle. He described a lot of psychoactive species, probably unknowingly. But he described that one, it was either from Cuba or Jamaica, I forget. I'm inclined to say Jamaica. He described it from Jamaica, consulting the North American Pluteus expert, Alfredo Justo. He believes this is the true Pluteus harrisii. Based on its geographical proximity to where it was originally described from, as well as my buddy John, he's made a good habit out of doing microscopy or on a lot of the collections he makes. And Alfredo said that this one, microscopically, does align better with what Merle called harrisii. So potentially, this is an example of one we're going to be able to resurrect, like this almost 100-year-old name for a Pluteus species that, you know, no one's used since Merle, and it turns out it is psychoactive. And here's a good example of the bluing halo at the apex of the site. So you can see this deeply pigmented blue on the species. And then I believe, you can even see a little bluing at the top of the stipe there. Unfortunately, this photo's upside down. Now, there are other Pluteus species that I've forgotten to add to this list. One of them was collected by my buddy JT Flanders here in southeast Georgia. He found what seems to be a novel Pluteus species, undescribed. Alfredo couldn't match it to any of the known named species. And then recently, there was confirmation through DNA sequencing of another one from Georgia, but this time the Atlanta area, Pluteus saupii, is another known Pluteus species, spelled Saupei. So I just forgot to include those. My apologies. Now, okay, this last one. This was the asterisk one. So Galerina species. A couple months ago, I was with my buddy Andrew Kunick as well as Ben Lemon, who's a PhD student at University of Florida. At least at the time, he's gotten his PhD now. And we came across these interesting LBMs growing on heavily decayed palm. And it was like covered in moss, and it was growing directly out of the palm wood as well as the moss. There were 3 fruiting bodies, and he noticed that they were bruising blue. And so he called me over, and he was like, "Hey, Scott, I've got some bruising blue over here." I was like, "Oh," and ran over, dropped what I was doing, and turns out it was a Galerina species. And by the time he had brought it over to me, 2 out of the 3 fruit bodies had fallen off and gotten lost in the leaf litter. So we were left with this one intact fruit body, and then it's hard to see, but there's a little aborted pin that was bruising blue as well. But with this one fruiting body, I made the most of it. I took some half-decent pictures. I also shone the UV light on it because I knew that a lot of Galerinas had these just spectacular UV reactions with all the Chylocystidea fluorescing. So I was able to get a nice photo of that, and then a photo of the dried specimen, which isn't very high resolution, but you can see this very darkening happening on the stem base. We did try and make a spore solution with this. We took a couple of gills, dropped it in water, and we tried putting it on a couple of petri dishes, but they never germinated. So this one we'll just have to find again. It has not yet been DNA sequenced, so I don't know where it falls phylogenetically yet, so it doesn't have one of these temporary codes, or who knows, maybe it's a named species that we just didn't know contained psilocybin. So that's why I have the little asterisk. Okay, it looks like I inserted the Galerina in the middle of the Pluteus section. So this is Pluteus saupii. So again, Atlanta, and then it's also found in other places. And then is anyone here familiar with this list? This is on the website, the Shroomery. This is curated by Alan Rockefeller. So it's a list. You can look up your state and see what psychoactive species grows in your state. So, for example, here's the one for Florida. I would take a lot of this with a grain of salt. Some of it is accurate, but there's a lot of inaccuracies. For instance, Alan has ovoids occurring in Florida. They've never been documented there. A lot of this is, speculative. Paniolus tropicalis was described from Africa on hippopotamus dung. It's never been confirmed in Florida. Combogeniensis. So it's I would say it's 80% accurate, but it's still a good resource. It gives you at least the genera to start targeting for your area. And then iNaturalist. I know I hammered it in throughout the talk a little bit, but again, before you post to Facebook or the Shroomery or wherever, Mycotopia, take some decent pictures, at least 3, get all the features of the mushroom, post it to iNat. If you don't want to go above and beyond and dry the fruit bodies and save them for DNA sequencing, if you at least take decent pictures, that'll attract people like myself, Alan Rockefeller, some of us hardcore mushroom nerds, to be like, what'd you find there? And we'll drive to where you found it and we'll collect it ourselves. but at the very least, high naturalist observation. It's a real life Pokedex. That's the best analogy I have for it. And at the very least, I live in Florida, and in South Florida we have all these tropical fruit trees. So I can go on and I can type in mango tree, and I can look at all the observations of the mango trees, and then I can go and steal people's mangoes or avocados, and it's great. So at the very least, you can use it to steal other people's food. It keeps track of all the observations you've made, where you've been, and one really cool I like about it is I'll forget about observations I made like a year or two ago, and I'll go back to them, and I'll be like, oh, that was such a good day. I found this really cool mushroom, and there's that river I swam in right next to it, and I camped out there, and I met these cool people here, and so it's kind of like a photo album that rekindles these lost memories. So it's really good for that. If you want to go above and beyond for science, right now, for those of you not familiar, there's a guy named Stephen Russell who lives in Michigan, and he's doing mass DNA barcoding of a ton of collections throughout the United States, and he has an open call for a lot of states in the U.S. And in the Southeast, he currently has an open call for Alabama and Florida. Unfortunately, Georgia, South Carolina, and North Carolina are not in there, Tennessee. But about 3 or 4 times a year, he also has an open call for the entire continent of North America, and it's limited to 10 collections, so he'll set up a window of time, usually a week to 2 weeks long. Anywhere in North America, your 10 best, your favorite collections, the most interesting stuff you've got, you can send it in for free to Stephen Russell for free DNA sequencing. And
when that happens? How do we know when that happens?
Stephen Russell pretty much strictly operates on Facebook. So, and then, whenever he posts that, a lot of the local mushroom clubs tend to share that information as well through Instagram, Facebook, whatever other social media things. But also, Stephen Russell does have a website called mycoda.org, and it's pretty up to date with it. He usually he's good at keeping it up to date, so you can get that information there as well as his list of open call states who can send in unlimited specimens. That's my talk. That's my name. Any questions?
That was so good.
Thank you.
Yeah.
Do you think that they're evolving at a rate that's too fast for us to figure them all out?
That is above my pay grade. I don't know. Josh, do you have any?
Almost certainly not. In most cases, because most of the time for sort of natural or the standard speciation processes that we think of, it's probably, I remember talking hundreds of thousands of years normally for that to happen, typically. However, there are certain other ways in which it can happen, which Diviotropicalis might be an example of. Be speciation through hybridization, which is the thing that does happen relatively instantaneously, as opposed to having to take that long scale time of like gradual accumulation of mutations. So occasionally you get speciation through hybridization. Even still, that's not something that probably happens like all the time. Not so fast that we can't keep up with it. In all these instances of these species described from the 70s, 80s, 90s, and today, is more finding stuff that already exists as opposed to sort of just immediately arose, with possibly a few exceptions. Some really recent speciation events.
By the way, Josh is an expert fungal taxonomist, PhD mycologist. Dusty, I mentioned him to you. He's in Tennessee.
Yeah, I was just putting that together.
I wanted you guys to, and expert on Clavariaceae.
Coral function. Coral function.
Yeah.
Nice.
Pretty ones.
Close.
Any other questions, concerns?
Is anything that turns blue when it bruises?
That's an excellent question. So this is like a hot tub jacuzzi thing. Not all mushrooms that bruise blue are psychoactive, and not all psychoactive mushrooms bruise blue. At least readily. So there's a lot of bolete mushrooms that have these crazy chemical reactions, including some blue staining, and they are not psychoactive, or at least psilocybin active. There are reports of an Asian species, I believe they think it's Landmoa asiatica, that is a blue bruising bolete that does not contain psilocybin. It's unknown what the profile is, but it tends to produce these anticholinergic effects similar to something like Datura or Brugmansia, but they still haven't gotten to the bottom of it. And I think the problem is that they've been approaching it from too scientific of a approach, where they're getting all these full genomes of like everything they can find at these markets in, I think it's Yunnan, China. But they haven't like bitten the bullet and started experimenting and bioassaying the mushrooms themselves. So they're like looking for this like little signal, but they don't really know what they're looking for. But and it seems as though like the knowledge behind which species it really is has been lost with like some of the native people in that area. And like I think it's also been reported in the, what's it called? I forget what, close to Yunnan. Yeah. Does that answer your question? Maybe too much.
A little.
There was a story in the news recently about a mushroom in China that makes one see elves. And I was wondering if
so that's the one I'm referring to.
the
yeah.
belief from China.
What do they call it? Like
And Janet Yellen took some
yeah. They call this
and saw elves.
yeah. There's a word they called it, like the diminutive
Fascinating.
diminutive hallucinations. It's hallucinations specifically of little people, which is a recurring theme in a lot of like mythologies around the world, which is very interesting.
Can we get a hold of that here in North America?
You know, that's a good question. It'd be interesting if they did get to the bottom of exactly what species that was, and we found out, you know, what genus and maybe what section or subgenus it was. Then we started comparing some of the closely related species we have in South or in North or even South America. Maybe we would find more of that. And that'd be pretty interesting.
Yeah.
Did you have another? Okay.
Where are all the morels right now?
Atlanta, Georgia. Is that it? No further questions?
It's not a question, but I'm in the Georgia area. I'm looking to find more people to kind of connect with and learn about mushrooms, foraging, and everything else. So if you guys want to meet up or exchange information, I'd love to kind of do that.
The guy who had the ovoid photos, Instagram handle FLeashRoom. If you want to take that,
Yeah.
I'll pull it up right now. He's really cool, super kind, super nice. He's more than willing to just like share all his spots and
Okay.
Okay, FLeashRoom. Yeah, and through him, I've met like a bunch of people that he tends to go out with. They're all very cool individuals.
I'm always looking for people to do that with, and
Yeah,
I've been finding more and more areas where I can go, but mushrooms really haven't been in the areas I've been at yet. At least this time of year.
well, yeah. I mean, as far as I can tell, right now the only things popping in the Atlanta area are morels, ovoids, and then there was some agrosity.
Okay.
But I literally saw nothing else. It's a little early, I guess.
Yeah.
All right,
All right.
that's a wrap.